Hello and welcome back to the NWR Virtual Healthcare Conference. My name's Matt Wright. For those that just joined the investment panel session that just wrapped up, we hope you got plenty out of that. Thanks to Mark at BioShares who hosted the session, and we'd encourage people who are interested in the sector and who joined that to have a look at attending the BioShares Summit in Hobart in late July this year. It's an excellent event, and I'm sure you will get plenty of excellent information out of that one as well. One of the companies that will be presenting at the conference in Hobart this year is up next, which is Arovella Therapeutics, listed under the ticker ALA. Joining us is the CEO and MD, Dr. Michael Baker. Before we get started, a reminder that if anyone in the audience has any questions, please feel free to type them in using the Q&A function within Zoom. I'll now pass it over to you, Michael. Please go ahead.
Great. Thank you very much, Matt, for the introduction. And of course, big thank you to everybody for taking time out of their schedules to listen in today. I am delighted to talk to you today about Arovella Therapeutics. And as Matt said, we're listed on the Australian Stock Exchange under the ticker ALA. And we're very excited by our invariant natural killer T cell platform, which I'm looking forward to telling you more about just now. And I'll just put up the disclaimer. I will be making forward-looking statements, so please do take note of that. And I'd just like to start here at the highlights of the company. So as I said, we're working on what we call an invariant natural killer T cell platform. And what that provides us with is an off-the-shelf iNKT cell platform, which we're using to treat a range of different cancers, those that produce CD19, so lymphomas and solid tumours, through a partnership with Imugene, and also DKK1 producing cancers. And we continue to work with world-leading partners, Imperial College London, where our iNKT cell platform was derived, and MD Anderson for our DKK1 targeting asset, and excited to have an ongoing strategic partnership with Imugene. Now we have built and continue to build a really strong leadership team. We're delighted to appoint that only last week we appointed Dr. Tom Duthie as chairman of the company, which is very exciting for us. In terms of the company and how we think about assets and clinical indications, we're very much data-driven, which we utilise to bring into our decision-making. And we'll continue to survey the landscape to look for strategic acquisitions for technologies so that we feel align with our current platform and our focus areas. And so for all these reasons, we do see that we are well positioned. We're in a niche area. We're the only ASX listed iNKT cell company, and one of a handful globally. And we think for all these reasons that we are positioned well for growth. Now, let me just dive straight into cell therapy. I'm sure many of you listening today have heard of it, but for those of you that haven't, it really has ushered in a revolution to how we think about cancer treatment. And that's largely because of the impact that it's having already. Now, it is more affectionately known as CAR-T. But for these particular products that were administered to patients experimentally now more than 10 years ago. So in February last year, we saw the culmination of a series of reports for adult cases. But also in May, we saw the culmination of some media reports for Emily Whitehead, the first pediatric patient to take this type of therapy, both 10 years cancer-free after taking those therapies. So we've seen great success, so much so that we're using the word cure and cancer in the same sentence, which is lovely. And for the first two CD19 approved therapies, Yescarta and Kymriah, one's just done over 1.1 billion in sales, the other just over 500 million in 2022, and another a BCMA targeting cell therapy that was also approved in 2021, just did under 400 million. So they're already generating strong sales, and we see that the market is predicted to reach quite high levels, just about 12.3 billion by 2030. So with all that said, as I said, it's a great, wonderful therapy, but there are challenges that continue to persist. And one of those really, it's manufacturing and logistics, and still poses a really big challenge for the sector. And that's because for these types of therapies that rely on T cells, they must actually take the starting material for the product from the patient. So what this ends up doing is creating a very logistically challenging, but also a very costly way of getting the therapy to people, because you must make the therapy for every single patient. So it's in the order of about $500,000 American dollars per patient. In addition to that, because you're deriving the therapy from the patient that has the particular form of cancer, they've probably been exposed to chemotherapy or radiotherapy So it stands to reason that the starting material may be compromised, which makes the product less efficacious. And of course, there's a limited number of centres that can actually do all this work, which means that not all the patients that are eligible to be treated with this type of therapy are able to get access to it. Now, in addition, it takes quite a long time to make the therapy for each person, somewhere in the order of 4 to 6 weeks. Now, that's something that groups are working hard to reduce, but for the moment, what can happen is patients that are actually eligible, and even while the product's being manufactured, they can die during manufacture. And of course, because you're making it every single time, it means that you've increased the risk of manufacturing run failures. So we believe that our allogeneic CAR-INKT platform does have the potential to solve these challenges, but also the great potential of being a more efficacious product as well. Now, what do we have exactly? And it's iNKT cells that I mentioned a moment ago. So they're a naturally occurring immune cell, like a T cell or an NK cell. But we know that there's a whole range of benefits that come from this particular cell type. Firstly, they've evolved to naturally target and kill certain cancer cells. And they do that through what's called an invariant T cell receptor on their surface, which engages with something called CD1D on various cancer types. Now, interestingly, unlike a T cell, a classic T cell, this receptor doesn't change between people and always recognizes the same target, which actually means that we can give them from a healthy person to a patient. So they don't cause what we refer to as graft versus host disease, and that's what enables us to use these therapies off the shelf. Now, in addition to that, iNKT cells now in the literature are well appreciated to shape the tumor microenvironment by impairing or blocking or killing various cells that actually promote a pro-tumorigenic environment. And they also cross-prime other components of the immune system like T cells and NK cells, which can then join in the fight against cancer. And so what we do is we engineer them with a chimeric antigen receptor, which is like a turbocharger, which helps guide them to find and kill the cancer cells. And we've got excellent data that demonstrates that when we do this, they mount a very rapid and robust response against tumor cells. So just taking that a step further, just looking through some more of the advantages, as I mentioned, relative to T cells and NK cells. The iNKT cells have multiple targeting mechanisms, as I mentioned, through either the CD1D directly killing cancer cells or by influencing the tumor microenvironment or other cancer cells, or other immune cells, I should say. They have both the T and NK cell mechanisms of killing. They naturally suppress graft versus host disease, which means we can use them off the shelf. And unlike T cells, we don't require any further genetic engineering to enable us to be able to use them off the shelf. So just to really make this point clear as well. So as I mentioned, I've talked through now T cells, which a lot of groups are working on, NK cells, and we're focused on the iNKT cells in the center here. And we do feel that this is a potential area that has been untapped. And if we make this relative to T cells, this is just a representation of the groups that are working on T cell based therapies. And same thing then for the NK cells. So you can see already it's very crowded. Whereas when we look at the iNKT space or the CAR-INKT cell space, you can see that there's a lot more room for us to move, and it is far less crowded at this stage. So in terms of the manufacturing, I highlighted that as a really important benefit for these types of cells. So let me just take you through what it would look like to manufacture this type of therapy. So the very first difference is we take the initial starting material from a healthy donor, and then we isolate the iNKT cells from that individual. We then reprogram them to produce the chimeric antigen receptor, which, remember, is like the missile that will drag these cells towards a cancer cell and then trigger its destruction. We then grow those cells up into billions before ultimately vialing those cells, and then when they're needed for the treatment arm, they can essentially be thawed out of the freezer and then given to the patients that are eligible to take the therapy. So major advantages here are, one, healthier starting material, because we're not taking the iNKT cells from somebody that's seen cancer or chemotherapy or radiotherapy. It is scalable, so we know that we can make more than N=1, which is really problematic for the T cell sector. So we can reach more patients and far more cost effectively, and we get faster access to patients who may have a particularly aggressive form of disease. And as I mentioned a moment ago, it does remove that risk of manufacturing run failure, because once the batch has been completed, it's undergone QC and QA. It will be in the freezer stored, ready for use. So in terms of our potential cancer targets that we're going after, we do have a number of shots on goal in our pipeline. We've got ALA-101, which has a CD19 targeting CAR, which we're using to tackle hematological malignancies like non-Hodgkin's lymphoma. And we're also partnered with Imugene and their onCARlytics platform, which is very clever in hijacking tumor cells to produce CD19. So we're going to use the combination to potentially tackle a range of solid tumors. We've also got our ALA-104, which we're utilizing to target the DKK1 peptide, which also opens access to other solid tumors and potentially multiple myeloma. So a really nice feature of the platform is that we can continue to screen for new chimeric antigen receptors, or CARs, that we could essentially use to target a range of different blood cancers or solid tumors, or even start to consider having dual CARs inside the iNKT cell platform. So in terms of our pipeline, just taking it a step further, we have ALA-101, which is our most advanced product. We're currently working through the manufacturing stages to take that into phase 1. With Imugene and ALA-101, we're working through the proof of concept stages already and getting some interesting data for that combination. And for DKK1, we're still currently validating the target and then looking to combine the chimeric antigen receptor with our iNKT cell platform. So I'd just like to talk a little bit more about the data, because it is quite compelling, and it illustrates why we're so excited by the platform and why we think there can be not just this notion of off the shelf, but also the potential for to have a more efficacious product. So looking here at ALA-101, what we've got here, it's an animal study where we use mice, and we're looking at the underside and the backside of the mice in the top 2 rows. And we have what we call day 0, which is before the animals have received any treatment, but we have engrafted them with a particular form of cancer that produces both CD19 and CD1D. And then on the bottom 2 rows, we're looking at mice that have been treated for 3 days with a range of different therapies. So in the first column, we have PBS, which is essentially untreated. We have unengineered T cells, unengineered iNKT cells, and probably the most important control here is the T cells that produce a chimeric antigen receptor that targets CD19. And of course, in the last column, it's Arovella's ALA-101. And so what we're looking at here on the bottom is anywhere you see color in the animals, that's actually the tumor cells that are present. And so when we look at the bottom 2 rows, we see that in all of the what we call control groups, you see that the color does still persist, which indicates the tumor cells are still there and quite abundant. And then that's in direct contrast to the animals that have been treated with the iNKT cell therapy. We see that there's a strong regression of the tumor cells. So very clear that after 3 days, we've had a very strong response. And so it's great that it works quickly, but what we need to know is, does it actually improve survival? Because that's really, at the end of the day, that's what we're trying to achieve in humans. So this is a study where we take a much larger number of animals, and then we take the study out for a much longer period of time. So no longer stopping at 3 days, which would be here. We're taking the animals out all the way to 90 days. And what we're looking at here, it's called a survival curve. And if 100% of the animals were to stay alive, the line would remain up the top. And if the animals all die, then the lines drop down to 0. So for the 3 true controls here, we see that all the animals have died between 40 to 60 days. And in the orange line, what we're looking at is actually the T cell treated animals with a CAR that targets CD19. So it's quite impressive data already, right? That we have 60% of animals alive at 90 days. But what's more impressive is the iNKT cells. We see that it's in the order of 90 to 95% of the animals that are still alive at that point. And so this is, again, a very exciting finding that came out of these studies, and something we will be continuing to follow up on. But let me just walk through this slowly. So we had 4 mice, so there's 4 groups, 1, 2, 3, 4. And we're looking at the same animal in each group of 4, but just over a number of days, ranging from in the order of 10 to 60 days. And so all 4 of these animals were treated with our therapy. And so initially, as we saw in those previous images, the animals looked like they'd completely eliminated the cancer. And what we saw between, let's say, 20 to 30 days later is the cancer started to come back into and really localized to the brain of these particular animals. And with no further dosing, the tumours are actually able to resolve a second time spontaneously. So that's quite a remarkable finding, and it does open up the possibility here that the cells are remaining long enough in order to be able to eradicate the tumours a second time. But it also suggests that they're able to tackle a more solid tumour-like tumour, such as what we see in the brain here, and also to cross the blood-brain barrier, which is quite interesting. So I'll switch gears now and just talk about the partnership with Imugene and Arovella, and where essentially it's a strategy to mark solid tumours with a particular target and then destroy them with the CAR-INKT cells. So I've just shown you ALA-101. It's very potent and rapidly activated to kill CD19 producing cancers. And we're currently developing that for an off-the-shelf product for cancer for non-Hodgkin's lymphoma. CF33, on the other hand, it's an oncolytic virus. And I'm sure now, off the back of COVID, most people are aware of particular viruses and the fact that you have the coronavirus, which affects respiratory cells specifically. Well, CF33 is also an oncolytic virus, or a virus that specifically targets tumor cells. And so that's really important for what we're trying to achieve, because we want it to go to tumor cells alone and not to healthy tissues. And taking it a step further, some more clever people at the City of Hope have engineered it a step further so that when it infects the tumor cells, it actually continues to grow and it forces them to produce CD19. So Imugene are making great steps already with that product. They've already commenced phase 1 for the virus alone, which gives great data, of course, about safety, also the distribution and so forth. And so looking at how this might look from a practical point of view. So what would happen is you would take the oncolytic virus, it would be injected into the patient that has the tumor. It's then able to go and infect the solid tumor cells, where it will naturally, its job will be to replicate and eventually burst some of those tumor cells. But as I mentioned, it will start to produce what we call CD19, and that's the little green bumps now out on the outside of that tumor cell. And so that's really important for the combination, because our CD19 targeting CAR-INKT cells will then be administered and be able to come over the top and essentially find those cells that have CD19 expressed and destroy them. Now, I mentioned earlier that INKT cells also impact the tumor microenvironment by destroying or blocking certain pro-tumorigenic cells, but they also cross-prime other immune cell components. So we see that by bringing the two components together, we really have a multi-pronged attack on treating solid tumors, which is quite exciting. And the research collaboration has already progressed from the in vitro stage, and then we're now moving into the in vivo testing based off promising results. So in summary, for that program, what it actually does initially for the CAR-INKT cells, or ALA-101, we were initially looking at just targeting blood cancers for that product, but by combining it with the oncolytic virus, we see that it helps us shine a light, or puts a lens onto other types of cancers that are also very particularly nasty. And of course, solid tumors make up about 90% of cancer diagnoses, so it also accounts for a huge market opportunity to tackle. And I'll just lastly touch on our ALA-104. So this is our monoclonal antibody that we licensed from MD Anderson. And as I mentioned, it's a cell therapy it will be a cell therapy that we're going to use to tackle multiple myeloma and potentially solid tumors. So typically in the literature, DKK1, it's got a job to do as what we call a negative regulator of the Wnt signaling pathway, which is now well appreciated to be impacted in various malignancies. This protein itself has also been shown to be overexpressed in numerous cancer types. And what we're actually targeting is the protein itself gets made inside of a cancer cell, and then it gets chopped up into small fragments, and then it gets loaded onto an immune complex, where it then gets shuttled out onto the surface of the cancer cell. And so that's what our technology will actually recognize, that particular peptide of this protein stuck on the immune complex. Now, it recognizes only HLA-A2, so that's a specific phenotypic marker, and approximately 40 to 50% of the population are what we call HLA-A2 positive. So we see that there's the potential for quite a broad role in numerous cancers, but also then potentially large market. So in terms of activity to date, we've demonstrated that the actual monoclonal antibody itself has activity against multiple myeloma cancers, as well as things like breast cancer. And then when we put the technology so that it becomes a chimeric antigen receptor in T cells. We've seen excellent data in multiple myeloma, pancreatic cancer, lung cancer, and breast cancer. And so the idea for us is to take the chimeric antigen receptor and insert that into our iNKT cell platform. Initially, what we'll be targeting is multiple myeloma because of the lower bar for blood malignancies. So just to give you a snapshot of the data that we've seen to date, as I said, this is now 3 different models, 3 different animal models of multiple myeloma. And we're looking at these survival curves where, again, 100% the animals alive will be at the top. And if they all die, it goes to the bottom. And what we see in the black line in all 3 of these experiments is that when the animals are treated with nothing, essentially they die between, say, 30 to 40 days. But in the case of the DKK1, and this is CAR-T cells, not iNKT cells, we see that they're all alive out to 50 to 60 days. So very compelling in terms of activity. Now, we also know that multiple myeloma is a form of blood cancer where a number of those cells will produce CD1D. So we do expect having the CAR inside iNKT cells could also make them dual targeting and potentially more effective. So in terms of preclinical safety, as I said, we're still validating the target, but to date, it's been shown that the CAR-T cells only kill the cells that have the target on their surface. They don't kill healthy blood cells or cause weight loss when given to mice. When we look at a range of 35 tissues, only 1 is non-specifically detected, which is the tonsil. And so as I said, we're continuing to confirm that the technology doesn't target or attack healthy cells, and in parallel, combining that with our iNKT cell platform. So we do have a very busy year ahead, and certainly off to a good start in 2023. But for our lead program, CD19, or CAR-19 iNKT cells, we're continuing to work through the manufacturing. So really in the next 6 to 18 months, we'd like to complete the clinical manufacturing, commence phase 1 for this particular product with non-Hodgkin's lymphoma patients. For the combination with Imugene, we've already made quick progress in terms of proof of concept studies. So we'll continue to advance those and move towards IND enabling studies for the combination therapy. And for the ALA-104, we'll continue to optimize the CAR structure as well as validate the target, getting ready for more IND enabling studies. So as I mentioned at the start of my the talk here, we do have a really strong team. It's remarkable, I think, the caliber of individuals that we do have in the group. It is a small group, but I think it's a very powerful group, and we do operate virtually, so we don't have bricks and mortar that we need to worry about. And as I mentioned at the start, we're very fortunate that Dr. Tom Duthie joined as the chairman at the beginning of last week, which is very exciting for the company moving forward. So in terms of the financials, we're currently just valued at about over 25 million. At the end of December, we had about $5.2 million cash in the bank, including a placement we completed in January. And we've got what we believe is an excellent register. Delighted to have the support of larger investors like Merchant, and we have a number of small institutional and high net worth investors on the register. And it'd be delighted to see a number of other investors join the register, and of course those that appreciate the risks, but also understand the returns that can come from an investment in a biotechnology company like Arovella. And so with that, I'd just like to summarize, as I hope I've illustrated, we do have a really exciting off-the-shelf iNKT cell platform. We're unique as a result of that. These CAR-INKT cells have multiple anti-cancer properties, I should say, through the chimeric antigen receptor, through their natural ability to target and kill cancer cells, as well as their ability to shape the other aspects of the immune system. Now, we know that once we get these cells manufactured, the allogeneic factor will significantly improve logistics and patient access, which is exciting. We continue to expand our pipeline with now ALA-101 and 104, and I didn't talk about the novel cytokine technology that we have an option agreement for with the University of North Carolina. But again, we will continue to look to evolve and expand. We continue to nurture our partnerships with really prestigious institutes like Imperial College, the MD Anderson Cancer Center, and delighted to continue to work with Imugene on the combination product. So as I mentioned, I do believe that we're in a very good position. We're unique, we're differentiated with a really exciting platform, and certainly looking forward to getting these products into clinic. And with that, I'd just like to say thanks very much again to NWR for putting the event on, and of course, thanks to everybody taking the time to listen in. Appreciate it. And I'd be happy to answer any questions.
Thanks, Michael. We will jump into some questions. Once again, if anyone would like to submit one, please do so using the Q&A panel within Zoom. The first one, Michael, is can you give some background on the work that went into bringing ALA-101 into the portfolio?
Yeah, absolutely. So that was it was an enormous amount of work. We spent probably 18 months scouring research institutes and universities across US, Europe, even Australia, until we found what we were looking for. And I think essentially it had to be something where we felt that we're at the cutting edge of oncology and cancer treatment, but at the same time, it needed to stand out. And so we did consider looking at T cell and NK cell products, but as I highlighted earlier in that slide where it shows they're quite busy, it was important for us to find something that stood out a little bit. And so the invariant natural killer T cells certainly hit all those boxes.
Very good. Another question is, can you speak to any key developments with similar technologies recently, whether it be in Australia or globally?
Yeah, absolutely. So there's, as I mentioned, there's the three companies that I would say are in our direct space. So there's MiNK. They recently put out some interesting data demonstrating that without engineering the iNKT cells, so they're just the native iNKT cells, that they could dose up to 1 billion cells without side effects, right? Which is great data for us to be able to leverage, and certainly paves the way for why we think they'll also be quite safe. Another group, Appia, which we don't know much about, they came out of stealth May 21, and back then it expected to be in the clinic by the end of this year, but we haven't heard too much. And then the last is Athenex, which is quite an interesting company. So they have also CAR-INKT cell products. Unfortunately, they got put on clinical hold for one of their products, which is quite different to what we're working on. It's a GD2 targeting product that is a really important unmet need going after neuroblastoma in pediatric cases. So unfortunately, they did have a patient death in their clinical trial, but we don't know all the details around that. But it doesn't look at high level. It doesn't necessarily look like it was related to the treatment itself, but unfortunately, it was an infection that individual suffered. So it's very sad for that program and product, because they are on a clinical hold now, and hopefully they can get out of that in 6 months. But as I said, there's not much other activity in the space, and so as I said, we're delighted and looking forward to getting ours into phase 1.
Very good, and not to put you on the spot a little bit, but I know last week you saw Professor Yuman Fong, and who was the founder of onCARlytics, present. And was there anything that you took out of that presentation that sort of changed your view, I guess, or had you thinking about things a bit differently after seeing him speak last week?
I think it just reaffirms it, right? So seeing a bit of a snapshot and Yuman's enthusiasm for talking about the distribution, where this virus can get to, where it can go, the number of different tumor types it can infect. I think it's a really exciting concept, right? That you've got these two, really remarkably different technologies potentially coming together synergistically. So, if it plays out the way we think it will, then I think it's got a lot of potential across a lot of different solid tumors, which would be remarkable.
Great. Well, that's all we've got time for. So, Michael, thanks very much for the presentation on Arovella today. We'll leave it there. And next up, we have Avecho Biotechnology, who will be on in 10 minutes from now. But thanks again, Michael.
Thanks, Matt. Appreciate it. Thank you.